Hello everyone, my name is Geert-Jan Huizing. I'm a PhD student at the UNSPSL and the Institut Pasteur in Paris, supervised by Gabriel Perret and Laura Cantini. In recent years, the profiling of multiple molecular layers from the same cells has become possible. And thus there's a growing need for computational methods that are able to learn from this complementary data, for example, gene expression and chromatin accessibility, or surface proteins, and to jointly analyze these complementary regulatory layers in order to gather biological insights. Mowgli is a new integrative non-negative factorization model with an optimal transport reconstruction loss that pushes the boundaries of linear models for single-cell multi-omics integration. What it does is that it embeds a cell's multi-omic profile into a low-dimensional space that has biological meaning, meaning that each dimension, each factor, corresponds to a specific biological signature. For this, we extend the Wasserstein non-negative matrix factorization framework to multi-view data, and more specifically to single-cell multi-omics data. Unlike a traditional Euclidean loss that you might want to use for non-negative matrix factorization, optimal transport allows you to take into account the relationships between genes, between peaks, between proteins, when evaluating the quality of the reconstruction. Meaning that instead of comparing gene by gene the data and the reconstruction, H times W, you can take into account the context to gather a more comprehensive view of the cell. The low-dimensional embedding W can be used for clustering, for example using Leiden or Louvain, and for visualization using UMAP or t-SNE. And the loading H can be used for biological interpretation of the individual factors, using gene set enrichments on the top genes for that factor, or motif enrichments on the top peaks of that factor, or looking for markers among the top proteins. We compare Mowgli to the state-of-the-art for several metrics of clustering and embedding performance across several modalities and tissues. We start with some control settings that illustrate changes that are typical of single-cell data, such as complementarity between omics. And there, Mowgli outperforms other methods according to all metrics in all 6 settings. For more complex real data, the performance of Mowgli is competitive with the state-of-the-art across data sets. Now, in this case, the benchmark might also be affected by the fact that it's difficult to get a reliable ground truth for real complex data. While the competition or the race might seem a bit tight in terms of clustering, what really sets apart Mowgli is its biological interpretability. We showcase this on a 3-omics DC dataset that is profiled for RNA, ATAC, and proteins for each cell. And we show that Mowgli detects factors that are specific to each cell type present in the dataset. For example, for CD8 T cells, you can see that Mowgli has 2 factors that have a high weight in CD8 T cells and a low weight in other cell types, meaning that these 2 factors are specific of CD8 T cells. By contrast, MOFA+, the leading linear single-cell multi-omics integration method, has no factor that is very specific of CD8 T cells, in the sense that it would have a high weight in CD8 T cells and a low weight elsewhere. Instead, it has many less cell type specific factors. And you would think that maybe this is only restricted to broad cell types, like CD8 or CD4 T cells. But we showcase that Mowgli's biological interpretability also applies to more specific subpopulations, such as CD8's effector memory T cells, as shown below. For this, we identify markers among the top genes and proteins. We perform gene set enrichment and motif enrichment, as described before. And we find that those different layers of regulation confirm our hypothesis of effector memory CD8 T cells. Now, the interesting fact here is that the top motifs have targets among the top genes, which suggests strong regulatory mechanisms. Now, if you want to try this out for yourself, There's a Python package that's very easy to install, pip install Mowgli. If you want to know more about how to apply it, you can visit our GitHub or read our preprint, paired single-cell multi-omics data integration with Mowgli. With this, I would like to thank my supervisors, Sarah and Gabrielle, as well as Ina, that helped me on this project. And thank you for your attention.